Okay, now here is the framework for doing a critique or an evaluation of a study, and we're talking about the handheld calculator study. Now we want you to be able to analyze the study for theory and stance and design and sampling and data collection in your discussion groups. We're going to go on and talk about this particular topic, the data analysis and results now, and we have a handout that we'd like to use to do that. So here's a handout that we've created. The idea is to review some of the key points for the article, but we're going to focus on the data analysis. So we have a discussion of the pre-test, post-test treatment and control group design. And if we scroll down here, we see that graphic. We just used the same graphic. The idea of sampling and how random selection plays a key role in this particular study. The measurement or data collection, in which we have a standardized test, and the validity and the reliability should be noted as you go into that part. And then we get to the data analysis, or what we do with the data.
And inferential statistics are the kinds of statistics we use in experiments and quasi-experiments. And this is different. As you remember, the descriptive statistics gave us a mean and a standard deviation. And these two calculations are the basis for any other statistics we'll do inferentially. Inferential statistics allow us to make inferences. They allow us to determine what we call statistical significance, which we'll talk about in a minute, and then based on that to make inferences from the sample to the population. That is, if what we found is true of the sample, can we say it's true of the entire population from which the sample is drawn? And even further, can we say it would be true of populations that are similar to the population from which the sample is drawn? And
one of the words that is I think I want to just make sure that we don't get too far is the idea that something from quantitative research is truth. That is it likely? We're trying to build an argument that what we find in experiments is likely to happen, and that's what statistical significance is. So
it doesn't mean it's extremely important.
Yeah,
it means simply, as we say, it tells the degree to which the results we get, the probability to which the results we get are due to chance or to the treatment itself. In other words, it answers the question, what is the probability that the results of the intervention are due to chance or sampling error? And we obviously want to get results that have a very high probability of being due to the treatment and a very low probability of being due to a problem in sampling or any kind of error, which brings us to the definition of probability, which is represented as P. And P tells us the probability that the results are due to sampling error or chance. We usually set a p-value of around less than 0.05, which means that the outcome has a low probability, that's less than 5%, of being due to chance or error. And it has a high probability of 95% or higher of being due to the treatment or the intervention itself.
So the idea of probability is there to help us strengthen our argument. The whole idea is if we have odds that these are of 20 to 1, that is that 95% chance that this is due to the treatment, those are pretty good odds or pretty good reasons for us to choose this particular treatment. So in the handheld calculators article, we have the intervention, and we're saying that the intervention with the handheld calculators was really quite substantial.
And we always say that all roads lead to P, that whatever we do is we try to get to P, and this is how we do it. In this article, we use a test of significance called the t-test. And the t-tests are used with no more than 2 groups. And the t-test for independent means analyzes the difference between 2 groups. And the correlated means analyzes the difference for the same group before and after a treatment. So in this case, we have 2 separate groups, treatment and control group, and we're using a t-test to compare independent means. So that's the first thing, is the test of significance. And then we get a t-statistic.
Well, we get a t-statistics by looking at the ratio of the impact of this intervention, that is the mean of the treatment group, minus the control group, which is business as usual. So we want to have something to compare to. So that's impact, and it's all a ratio. It's also then is a ratio divided by the sources of error in a study.
Now, if you don't remember all that, the way the t-statistic is found is using a computer program that calculates it. And the t-statistic will yield a number. Usually it'll be a whole number followed by a decimal. And this actually is calculated from the data, as Jeff said, for mean and standard deviation to correct for any errors. So now you have a t-value. And then you need to get a t-critical value.
The t-statistic is from the experiment itself, so we use the means and standard deviations. The t-critical value is calculated from a series of studies, of hypothetical studies that will include the studies of different sample sizes and studies with different means and standard deviations. That is, a whole variety of comparable studies, but with different sample sizes and group sizes. So it's something to compare to. This is where the that normal curve comes in again, where we're comparing it to a hypothetical t-critical value. So what we're going to do then is compare the t-statistic that we get from our study to the t-critical value that comes from a table of t-critical values.
So just to go through the steps one more time. The first thing that's going to happen is calculating the t-statistic, and that's done through a formula that looks at standard deviation, which has to do with the sample, and means, which has to do with the scores. The next thing you want to know is what's the p-value you're going to use. Usually it's 0.05 or less. We call that the alpha. Then you go to a table called critical t-values. And on this table, you look down the column that has the p-value. So you go down the column that has 0.05, and you look at your t-value, and you see if your t-value is higher than what they call the critical value that appears in that column. If your t-value is higher than the, quote, t-critical value, then you've known that, given the size of the sample, you've reached the p-value you want. So it's a multi-stage process. First, calculating the t-value, figuring out what you're going to accept as probability, it's usually 0.05, then going to the table and looking to see what t-value you need to meet the standard of the probability you set. And then it's statistically significant if it meets the t-critical value or is above. And that's how statistical significance is calculated. You will later on, we'll talk about other kinds of tests of statistical significance, primarily something called analysis of variance, but the same procedure is always followed. You calculate the value of the statistic, you go to a table of critical values once you've figured out what kind of probability you want, and you use that table to determine whether or not the statistic you got meets the standard of the probability that you've set. And that's how we know that you've reached statistical significance.
And I think you'll agree on the one hand that this is all laid out and very objective. That is, there are no changes, there are no variations on this. This is the procedure you follow. Even if you think this is a little overwhelming and complicated, I think you'll agree that this is an effort to try to make this as objective and as understood a process as possible.